So this is Dr. David Williams, who's going to be speaking next. Dr. Williams is the chief of the division of Hem/Onc at Boston Children's Hospital and the president of Dana-Farber Children's Cancer Center, and the associate chair of oncology at Dana-Farber Cancer Institute. So this is one of the leading oncology and transplant institutes in the world. He's going to be speaking to us on gene therapy vectors, and we're very honored to have you here, David, so thank you for speaking.
Thank you very much, and I apologize in advance for having to depart quickly, but I really appreciate the invitation to come and talk to the community. And so thanks to the organizers for inviting me. So as is typical for academic presentations, these are my disclosures. There's nothing here that's relevant to the talk today, except that I have served on the advisory board for Bluebird Bio for their FDA applications. So just to set the stage for our talk, I'll just remind you why it is that we use hematopoietic stem cells to treat neurologic conditions. So remember that ALD causes an increase in very long-chain fatty acids, and these are particularly damaging to cells in the central nervous system, and particularly to myelin, and to the cells that repair myelin, you can see on the right, including glial cells that I'll talk about more in a moment. And so why do we use hematopoietic stem cells to treat the disease? This is secondhand to many of you, but some of you may not know this. So you can see in this diagram that derived from the blood stem cell, which resides in your bone marrow, there's a type of cell called a myeloid cell or macrophage that we believe migrates into the brain and differentiates or becomes microglia cells. So these, when we use gene therapy, now carry a normal ALD gene or ABCD1 gene, and this replaces the damaged microglia that are in the brain not doing what they're supposed to be doing, which is repairing the damage to the myelin. Okay, so I had a little movie. I'm going to skip the movie for the sake of time and talk a little bit now about the trials that have gone on previously, most of which has been published. So for gene therapy, remember what we do is we collect stem cells from the blood rather than the bone marrow in a process called apheresis. And that's in a machine you can see there next to the patient. And those cells are taken to a good manufacturing process lab. The FDA considers this a medicine, so it's all handled in FDA regulatory fashion. And the gene transfer, you can see on the bottom box, occurs in this case with a virus vector called lenti-D. And then once all the safety testing is done, release testing is done in the GMP lab, the individual who's getting the gene therapy is admitted to the hospital. In this case, they received conditioning. In the first trial, they received busulfan and Cytoxan, and in the second trial, they received busulfan and fludarabine. These are characteristic chemotherapy agents used to do bone marrow transplants. And that then is infused. This is one of the things that has fascinated me for my entire career. The blood cells the stem cells are infused in the blood. They know how to get into the bone marrow. They know how to arrange themselves in the bone marrow so that they now can produce all the blood elements that then circulate. And some fraction of those circulate into the brain and become these microglial cells, and then these cells then stop the progression of the ALD disease. So this is, again, published recently data. This just shows you in the two trials combined the overall survival. So the two trials have different colors, but you can see the overall survival has been quite good in these stem cell gene therapy studies. But you know probably already very well that that's not the end of the story. So now you see that this is event-free survival, which is the way a scientists think about these trials, and survival free of major functional disabilities, neurologic disabilities. And you can see when everything's combined, that's the top purple, you have relatively good outcomes. You can see that in the bottom kind of yellowish curve, the second trial called ALD 104 really drops off, and that drops off because of the occurrence of a complication of this trial, which is called insertional mutagenesis. And that's looked at in a different plot here, where the incidence of leukemia or hematologic cancer, which is the term that was used in the New England Journal papers, is shown. And you can see again in the ALD 104 that there's more of an instance of that versus the first trial, 102, even though the first trial occurred earlier, much earlier, and so the follow-up time is longer. And so this is the most up-to-date data that we have from the company, which is now called Genetics Biotherapeutics, not Bluebird. And you can see in the 2 total trials, there's now been 10 instances of boys developing either myelodysplastic syndrome, that's a fancy term for pre-leukemia, or leukemia. And most of these, 8 of the 10 occurred in the second trial, only 2 occurred in the first trial. So what's that done for the field? It's very problematic, of course, even though the treatment is effective, it has a serious adverse event. And this, just for the context, since I've been in the gene therapy field for a long time, this is very similar to what happened in the first gene therapy trials in the bubble boy disease called SCID. It was 20 patients, 10 in Paris, 10 in London. It worked very well, but 6 of the 20 boys developed leukemia from the same kind of complication. So what's happened recently is the FDA has changed the label for this drug so that now the indication is without an available HLA-matched donor for allogeneic. So not, if you have even an unmatched, highly matched donor, you're no longer eligible for getting this treatment. So let's take a minute and talk about why this occurred and what it means going forward. So this is most likely not because of the lentivirus, but more likely because of this specific lentivirus. And I'll make a point about that a little bit later on. So the picture at the bottom is how we, as scientists, show a lentivirus. You can see, for the purposes of this, in the blue box in the middle is the gene that we're trying to fix, called ABCD1. And I'll come back to this structure in a minute to go into more detail. But the point I want to make is that the gold box next to the blue one I'm going to drill down on is what drives the expression of the gene in the tissues once the gene is inserted into the tissues. So we need then to talk about gene insertion, and that's the process by which gene therapy actually works. And you can see here, I've pointed out again in the blue box, that's the healthy gene that we're trying to put into the cells that we need to. And it shows here that the healthy gene gets put into the genomes of all the cells that are derived from the hematopoietic stem cell. And that makes then, in the appropriate place, that makes the protein that's defective in boys that have this disease. Okay, so this sequence that I'm talking about is called an enhancer and promoter, enhancer promoter. So what is an enhancer promoter? So there's several definitions, but these are two that I like. So just to put into the context, the gene itself, so the ABCD1 gene represents the basic instructions to make a protein. The promoter is like in an orchestra, the band leader. So that's a small switch to the right of the instrument that signals when it's to start to play. But in the normal circumstance, that playing is at a very low volume. So not much sound is made. The enhancer is like a spotlight that looks on that promoter and says, turn up your volume, tells the instrument to make more volume. And that doesn't have to be located actually right next to the gene itself. It can be located way away from it. So the other definition is the enhancer is like a dimmer switch for the light. So the dimmer switch is located away from the light bulb itself. You have these in your houses, but it can control the brightness of that. So the enhancer, which is located far away from the target gene in the DNA, tells that gene to turn up its activity. So now you've gone through Molecular Biology 101. And there'll be a test at the end of the talk. No, I'm just kidding. Okay, so what has then gone wrong? It's called insertional oncogenesis. So here's the picture again. There's the ALD gene in gold. And here's the insertion of the gene. And that insertion either can decrease the function of the gene where the vector went in, or in our case, it can increase the expression and activate the gene. And if that gene is not supposed to be activated, then that's a bad outcome. And so here, this is complicated, I'm going to go through it a little bit. So at the top is our vector schematically again. And you can see again the ABCD1 gene in blue. And you can see this promoter enhancer with the little arrow. And down below is the genome of an infamous gene called MECOM. And it has two parts, MDS1, that stands for myelodysplastic syndrome 1, and EVI1, which stands for ecotropic vector insertion site 1. This gene was originally found in mouse genome to harbor insertions of viruses that led to leukemia in the mouse. And we know as hematologists in pediatrics that we see children develop leukemia because they have mutations that arise in this gene. Now down below is a fine-tuned diagram. And these numbers, ISC1, IS2, IS3, IS4, those are the exact sites in the gene where the first 4 patients who developed myelodysplastic syndrome, where the vector landed. And when the vector landed there, it inappropriately turned on the EVI gene. I'll come back to this as an important thing in a minute, but that's essentially what went wrong. And when this EVI gene was turned on inappropriately, it led to abnormalities in the stem cell, which subsequently, with additional mutations, led to the leukemias in these boys. This is known to happen. As I said, I mentioned already, we were part of the team that worked out the problem in the SCID trial 20-some years ago, and we developed a way to try to develop safer vectors, which we went on to do and test and have shown to be. But on the top row with the blue arrow, this is what's happening. It's called enhancer insertion. In the SCID trial, the gene was LMO2, another oncogene. In our trial, it's MECOM or EVI1. So why in the world did Bluebird 15, 16, 17 years ago, that's how long it takes, pick this vector with this MND enhancer? I think you can appropriately ask that. So at the time the trial was designed, it wasn't really known what cell type needed to actually express the correcting protein to stop the disease from progressing. And so the logic was, well, let's use a very strong promoter enhancer to make sure it's expressed in every cell. That way we don't have to worry about it. And so the MND-U3 promoter actually does do that. It expresses in many different cell types. And The enhancer was used in the first ALD academic trials, 3 boys treated in Paris, and that seemed to work without any problems. And before the FDA approval of the trial, Bluebird did a number of safety tests in vitro, and none of those tests actually indicated that there was any problem with this vector. And finally, there's a trial from my good friend and colleague Don Cohen at UCLA in the bubble boy disease with this same vector configuration. Those boys are now out over 15 years, and none of them have had a problem. Now, there was only 8 boys, so it's a small number. But all these things kind of went into the thought process. And the other thought process that was very important at the time was, you don't want to do this kind of a trial and at the end have not enough expression to stop the disease. And so the decision was made, and I was at those meetings almost 20 years ago. I think, Florian, you may have been too. The decision was made, well, if we have to decide, and we have no way of deciding other than these safety issues, we'll decide for the benefit of the patient, understanding there's a risk involved. Okay, so how do we see this in the patient's blood? So again, a complicated diagram here, and I'll just say to you those lines that go up, because the vector inserts and we can track, it's like a footprint or fingerprint, we can track the exact cells that have that vector in that location. This can be tracked at the molecular level to say, hey, wait, there's a group of cells that are growing with this insertion that are growing bigger than they should grow. And so this is called clonal dominance, and the diagram here is from 4 different patients that had that happen. And the point here is you can follow that prospectively and look for trouble evolving. And so Bluebird, now called Genetics Bio, continues to follow the clonal landscape longitudinally on all patients that were treated in the 2 trials, and now patients that are treated with the commercial product, Skysona. Okay, so is it the lentivirus? Because when this all occurred, there were a lot of people in the field that says lentiviruses aren't safe. This is from Bluebird's own database, and it's a lot of pictures or dots you can see. But I've bolded MECOM and another oncogene, PRMD16. And if you can tell, there's a group of gold dots that are high above everything else in that group. Those are the ALD vector patients, and all the rest of the dots are from sickle cell patients or thalassemia patients. Same lentivirus backbone, but a different promoter. So you can see that there's something about this particular vector, not lentiviruses, but this particular vector that's problematic. Okay, so what are we doing to treat the boys with this disease who developed this MDS? One is with the FDA, the company's developed enhanced screening, so monthly CBCs for the first year, a bone marrow at 6 months and 1 year, with what's called vector copy number, looking for how many cells have the vector in them at the same intervals. And then CBCs and vector copy numbers every 4 months in the second year, and then after that, every 6 months. Of course, determining whether, when this occurs, whether the boys have an alternative donor, and if there is excessive blast, that is, if leukemia has already occurred, then the boys undergo some chemotherapy before they have their transplants. And what are we doing to try to enhance the safety of the vectors? So we're testing new vector designs. And in our laboratory, that's a multi-investigator international collaboration, as we did with SCID, with severe combined immunodeficiency. It involves Mass General, Boston Children's, and the University of Padua in Italy. And the timeline for doing this preclinical work, we think, is probably around a year to 18 months. And the subsequent, what we call IND-enabling testing, which is the test with the actual material that we would then want to take to the clinic, depends now on funding. We have to find funding to do that. We're also developing new approaches using what's called gene editing. That doesn't use a viral vector at all. That's another collaboration with Mass General, the Broad Institute in Cambridge, and Boston Children's. And we've recently requested a very large amount of funding from a special process at the NIH. Of course, there's no one at the NIH right now, but it's the Thrive program. It's called ARPA-H. So we've asked them for enough funds to develop gene editing to go into the clinic. So let me just show you a couple pieces of data. I'm almost done, I think, time-wise. So here's two vectors that we've already started, we built and already started testing. You see the ABCD1 gene in purple. Now the MND promoter enhancer has been replaced on the top with a gene called PGK, which has been used in many previous gene therapy trials safely. And in the bottom is a new approach called the HLA-DRA promoter enhancer. The top one expresses at reasonable levels, but doesn't have what we call an enhancer activity. It doesn't activate genes far away from it. It just activates the gene right next to it. And the bottom one is interesting because it has low-level expression until it sees inflammation, and then it turns on. And if you remember my first diagram, what happens in boys' brains that have ALD is they get inflammation in the brain. And so presumably if this worked, this gene would turn on in response actually to that activation. So the diagram is back again, the 4 integration sites that turn on the gene. And on the right, you see our test vectors. And we've used gene editing to actually put those vectors in the exact spots that were previously causing the problem. And in the bottom right, you see the data. So on the first panel is PGK, that's black. The next one is a very strong enhancer that has caused leukemias in the past and other trials called SFFB. Then MND, that's the current vector. Then the HLA one I talked about in blue. And then the last one is MND in the reverse orientation. And you can see the dramatic reduction in the activation of the oncogene using these new test vectors. Okay, then let me end with gene editing. This is a new technology. You probably heard or may have heard of CRISPR. How does it work? You don't use a virus in this case. You have a sequence that targets a particular gene that you want to target. In this case, it would be the ABCD1 gene. This targeting part carries molecular scissors into the DNA. And so when that occurs, the molecular scissors cut the DNA, and it's specific at a particular point in the genome that you want it to cut at. And then in our case, when that cut occurs, the ALD gene would be inserted into that location. So the advantage of this is that the insertion is not random in the genome. It's only in the place where ALD is normally located. And the expression of the gene is dependent upon the normal expression control of the gene, the ALD gene. So those are potential advantages. There's other potential disadvantages, but this is certainly something that we're currently working on. So let me stop there. I know that there may be questions. I hope there are. I just want to make the comment to you as a community. It's amazing what you've done and what you've supported. And there's lots of people listed here, but the most important people are listed are you and the families that have supported this. But this has been a wonderful international collaboration of many different sites. And the fact that almost 70 boys were treated in these 2 trials is really amazing for a rare disease. It's just almost unheard of, actually. So I just want to express the appreciation of all of us researchers that work in this area. And thank you for your attention, and I'm happy to answer any questions.
So, David, you're sure you want to answer questions with
Yeah.
time? Is that okay? Okay.
Let me take the time to answer questions. It'll be fine.
Hi, thank you. In the case of gene editing, what's the delivery method? Is it a liquid nanoparticle, or is
Yeah.
it
It's
mRNA, or-
generally what's evolving. There's several ways of doing it, but what's really evolving into the clinic is lipid nanoparticles. And some of those are being dressed up so they go to a certain cell, but it's not very good at this point. But this is already into the clinic, yeah. And you may know this is sort of how the COVID vaccine was developed, is with lipid nanoparticles carrying messenger RNA into the cell, into the body. Yeah.
Thanks for the talk. Has anyone ever tried the ABCD1 promoter as a way to drive ABCD1? I mean, it seems like that's the promoter that's evolved to drive this gene.
Yeah, it's a good question. That's what the gene editing takes advantage of, because you place the gene, the cDNA, next to the cis-regulatory elements, the promoter and enhancer of the endogenous gene. It turns out the endogenous gene promoter enhancer are complicated and doesn't fit into a viral vector in a very efficient way. So this is sort of why it hasn't been. In some gene therapy trials, it is the endogenous promoter used, but most don't use that.
Yeah, thank you for a great talk. I'm trying to understand still, and maybe you did explain it and I just didn't understand it, but so when you did the experiment towards the end where you tried different promoters and inserted them into the region of the MDS1, why do you think is the MNDU driving this more than the other ones? Is it just the potency, you think, that it's?
Yeah, the ones that we tested, so there's the promoter itself, and then there's the enhancer element. The enhancer element turns things up. And the ones we're testing have very little enhancer activity. So they can drive the promoter, the gene next to them with the promoter, but they don't drive expression of genes that are far away. And that, in data I won't show you, it's a long time ago, we proved in humans that enhancer in the vector, if you leave it in, it causes these problems of overexpression of genes in the neighborhood, basically. So the question for the new vectors, of course, will be, okay, they're safe, they look safer, but are they going to express enough of the gene to stop the disease from progressing? And since we don't have great animal models for that, that'll be an issue that we have to address in the best way we can. But in the end, probably it'll be a clinical trial to determine that.
Yeah, wonderful talk. I have a super technical question. Reading some notes from the FDA, they seem to frown upon the woodchuck promoter sequence at the end of these constructs and have recommended moving away from that native sequence. And
Yeah.
the labs have made mutated versions. I know it's super technical, but do you have thoughts on that?
Yeah, in the vector, so we, as you know, we run many gene therapy trials that target the stem cell. We've modified that element in all of our vectors to the quote-unquote safe version. Yeah, it's a good question.
Yeah. So thank you again for a really nice talk. And I'm a big advocate for trying to make the viral vector approach safer to avoid insertional mutagenesis, and also at the same time trying new approaches like hopefully LNPs will find a way beyond the liver. But when you're talking about gene editing, you also risk genomic instability, so that may be a different problem than insertional mutagenesis.
Yeah.
Have you thought about potentially doing epigenetic editing, which is safer, and upregulate the compensatory gene ABCD2? Like, would that be even safer potentially?
Yeah, it's a good question. I think we're trying to do things a step at a time. I think one thing you're implying, which I would agree with, is that we really don't know completely the adverse events that may occur with gene editing, because few people have ever had, there are FDA-approved products now with gene editing, but it's based on very few patients and very little follow-up. So I think we'll be exploring multiple different approaches going in the forward, but these are the two we're doing right now, basically. Okay.
David, we want to thank you so much
Thanks.
for your time. And
Thank you very much for being here.